Good day. My name is Rutger Ploeg and I'm a transplant surgeon and professor of transplant biology at the University of Oxford. First of all, let me thank the Surgical Research Society and its past president, Professor Michael Nicholson from Cambridge, for the kind invitation. I'm honored to give the BJS Lecture 2025. As the Surgical Research Society concerns many young and enthusiastic academic surgeons who are very dedicated towards their career as clinician scientists, I thought it would be helpful to take them on a journey about curiosity, commitment, and collaboration. During my journey, I've had the opportunity to witness and participate in a few exciting milestones in transplantation research, and I hope that this will be of interest for you. It started all in Leiden when I decided to study medicine, which of course, as we all know, is very serious business, especially in Leiden, but may come with the occasional distraction. Nevertheless, it came to a good end, and at a certain point, my first impressions of transplantation were through Eurotransplant. Eurotransplant was a then very new organization, only a few years old, in 1969, founded by Professor Jon van Rood, who was an internist and immunologist at Leiden University, and one of the founders of the HLA system, the HLA tissue typing and matching to better allocate organs, improving survival. And his idea was to kind of go beyond national frontiers and exchange organs internationally, and that's why he set up this system, which was called Eurotransplant, which basically covered 8 different countries. And for us as students in Leiden, we had the opportunity to actually participate in this as a job, which was quite exciting, and it paid actually quite well, where we would doing off hours and in weekends exchange organs across borders. So, for example, a kidney that was allocated from Munich to Amsterdam, or a liver that was retrieved in Brussels and finally transplanted in Vienna. We did this, of course, the old-fashioned way, as you can see in the slide, with a so-called fax and telephone, if you still recognize those items. During one of the annual meetings, I saw Professor Khan give a wonderful talk, and I thought, well, without guts, no glory. So I approached him and said, well, would it be possible maybe to do an elective with him once I've finished my medical studies? And to my great surprise, and of course, very happy about that, he said yes. So the next step was then a clinical elective in lovely Cambridge. You see here the slides of the old Edinburghs, which now is a humongous bioscience park. And it was there during this clinical elective in Cambridge that I experienced for the first time sessions of liver transplantation, which at that time was a grand theatrical drama supported by symphonies of Richard Strauss and Gustav Mahler, since Professor Khan loved to have wonderful classical music when he was operating on the liver. What was the next step? Well, of course, I then had decided to want to go into general surgery and needed a specialist training spot, which came up at the Deanery of Leiden University Medical Center. However, in those years, there was no slot. I had to wait a little bit before the next one became open, and I took then the job as a first-generation transplant coordinator, which is kind of a Dutch specialist nurse in organ donation. So we had to set up the organ donation system in the western part of the Netherlands, assist in donor management, and coordinate organ retrieval for the regions that were affiliated with the Leiden and Rotterdam University medical centers. Obviously, at that time, a very new job, very exciting, and a great learning experience for somebody at that age. Halfway in my surgical training, I had 2 wonderful opportunities. First, to do a 2-year research fellowship at the University of Wisconsin at Madison, which is near Chicago, and one of the big 10 universities in the United States. And later, to be asked to come back and do my ASTS clinical transplant fellowship after completion of my surgical training. Madison is quite a a big research center and also transplant center with about 350 kidneys, 100 livers, 70 pancreases, and 150 retrievals at that time. Here you see Dr. Belzer, who was the head of the Department of Surgery, trained in Boston, then moved to San Francisco, became head of transplantation there, and finally ended as head of surgery in Madison, Wisconsin. Here's the early morning rounds, typically American at 7 a.m. And of course, all listening to the big boss. Now, Belzer was a very interesting character. He was really one of the true pioneers in organ preservation, and it doesn't go that far back as to Cosmos and Damien, but quite far. Let's go back to the first milestones in transplantation. We all remember, of course, the famous transplant between the Herrick brothers, Richard and Ronald, who were twins with a median function and a long-term success in kidney transplantation. Now, of course, this was a living donor transplantation and no preservation was necessary. And as both individuals were identical twins, not even immunosuppression was necessary. The donor and recipient were next door. So in that sense, there was no preservation since the kidney was retrieved and only 45 minutes later, it was transplanted into the twin brother. Now, of course, when deceased donation started, this whole scenery changed. And now suddenly organs were retrieved in different hospitals who were not even transplant centers, and the organs had to be transported back to the recipient center to either transplant the kidney or the liver or the heart or whatever organ. In the 60s, Belzer thought, well, we have to do something to better bridge organs from donor hospital to recipient transplantation. So why don't we simulate the situation as in humans? We need perfusion of the organ. And of course, since we want to reduce metabolism, why not do it in the cold? So he built this first Belzer machines, which you can see here, huge machine. It was portable, you see the wheels, but at the same time, only very small cassettes on top where, in this case, 2 kidneys could be transported from one place to the other. Now, in those days, in Detroit, the organ industry was still very active and also quite wealthy, so they had sympathy with Dr. Belzer, and Ford Motor Company actually built a special car around the machine, as you can see, to transport organs from A to B. So the basic concept in organ preservation, as I just said, became hypothermia between 0 and 10 degrees Celsius. So first you wash out the blood, you remove it, and then rapidly cool surface and also intravascularly to suspend the organ then in the cold and achieve a reduced metabolism, which hopefully then prevents ischemic injury. Following these first developments of machine preservation, new solutions came, Collins solution, Marshall solutions, and static cold storage, so without a machine, just in the bag on melting ice, became the norm, using much more simpler solutions than machine preservation, which was considered to be a bit complex. However, hypothermia comes with 4 classic unwanted effects. There's cell swelling, there's impaired energy metabolism, acidosis, and also precursors of reactive oxygen species, so-called reperfusion injury once the organ is reperfused and oxygenated. So the choice of components in those cold storage solutions is quite crucial to counteract those unwanted effects, which we call the solution effect. If we go back to cellular metabolism for a moment, under normal and hypothermic conditions, here's the typical normal cell function at normal thermia, the nucleus, the cell, the cytosol, high in potassium. There's the sodium outside the cell. There's the pumps, which are ATP driven, and the ATP obviously is produced in the mitochondria. And across the cell membrane, there's this so-called Donan equilibrium, which kind of keeps the sodium out by keeping the pumps active and ensuring that no sodium enters the cell. However, what happens then during hypoxia or hypothermia, like in organ preservation, basically the pumps slow down because there's less efficient production of ATP. And due to the fact that the pumps slow down, sodium can enter the cell, and with the sodium comes water, which results then in edema and in the end also in lysis of the cell. So how can we then achieve an optimal physical environment? And that is actually created by those agents in a preservation solution that counteract cell swelling and buffering. And there's like 3 good examples of that. There's the so-called osmotic pressure, which is given by impermeance, basically trying to kind of prevent permeation of sodium and water into the cell. And those are saccharides, for example, glucose being the smallest molecule, then mannitol, sucrose, much bigger, and then finally a large sugar, which is raffinose. It can also be achieved by the use of anions, lactobionate, phosphate, gluconate, citrate, and a certain composition of electrolytes, sodium, potassium balance, and histidine. You can also add oncotic pressure, which kind of keeps the intravascular compartment dry and then prevents edema from intravascularly going into parenchymal. And you can use for that albumin, hydroxyalpha starch, polyethylene glycol, or dextrans, which are very large molecules. And of course, the typical buffers used in preservation are, for example, phosphate, but also histidine. Now, there was a need for a better cold storage solution for all organs, but especially for the pancreas in those days, because there was a high risk of pancreatitis and thrombosis. At the same time, there was also an increased national and international organ exchange, and that longer preservation times were necessary. Now, this is the typical picture of one of the pioneers in transplantation, Professor Thomas Starzl, in one of his planes flying back from east to west coast with a liver on board, trying to reduce cold ischemia times. And this is the typical desirable preference of Dr. Belzer, waiting comfortably at home for organs to arrive. The real reason why, in the end, UW solution was invented. So when I came to Wisconsin for my fellowship, I kind of had the luck to kind of be able to participate in this development of a new cold storage solution, which we at those days called the lactobionate solution, because one of the major components was this impermeant lactobionate. The lab, Belzer's lab, was led by Jim Southard, who was our mentor and teacher, and we were the first research fellows who actually worked on the different components of the solution. There was Jan Warburg, who was very much focused then on the pancreas. Then I came focusing on the kidney, and then liver. It was Neville Jameson from Cambridge. And as you can see, the solution became much more complex than previous solutions were. But there was a good reason for that, because there was very much a systematic approach where we tested all components. Here you see, for example, that we looked for what is the best impermeant to kind of keep the edema out of the cell. And we tested different controls like Collins as an existing machine preservation solution, but then used components like gluconate or lactobionate and can combine them with sugars to see how the tissue edema and liver slices was able to reduce by the addition of those impermeants. And as you can see, the best combination was lactobionate as an anion and the raffinose, which was the biggest sugar. So those then became 2 components in the solution. Another good example was, for example, we want to prevent free radical damage at time of reperfusion. So why not adding an antioxidant into the solution? And glutathione was a very appropriate one, being very much natural tripeptide in all mammalian cells. It protects the energy regeneration capacity, and it helps to restore ATP. And as you can see here from this slide, is that we checked in renal tubules 48 hours of UW cold storage preservation plus reperfusion, and then looked at the ATP regeneration. And without GSH, glutathione, a less, let's say, an inferior accumulation of ATP versus when glutathione was used. When we moved this then to the transplant model in the dog kidneys, in those days we didn't do pigs but still dogs, you saw that a significantly better recovery of the kidney was possible when glutathione was added to the solution. So in this way, we tried to kind of find the effective components in the UW solution. And when you look at the previous solutions like Eurocollins, phosphate buffer sucrose, or Marshall solutions, HDK, but also UW were much more complex, however, also more effective in their organ preservation activity. And at the end of the day, we were able then in the 86 and 87 to come up with a new high-quality cold storage solution in multi-organ retrieval, where Jan Wolberg presented the pancreas data, Neville the preservation of the canine liver, being very successful for 24 to 48 hours using UW solution, and I had focused on the long-term storage of the kidneys. Coming back to Europe then, I thought it was the moment to try and introduce UW solution also clinically, and we started with help of the Dutch Kidney Foundation and Eurotransplant, a European multi-center trial in clinical preservation. We did this in kidney, liver, and pancreas. The kidney trial was very successful, as were the others as well. I just show you here the data. Dialysis requirement reduced significantly with introduction of UW solution versus then gold standard Eurocollins. And also graft survival was significantly better when UW solution had been used for the organ preservation period. So in the end of the day, after all these clinical trials in kidney, liver, and pancreas, a win-win in 91, when there was basically a global introduction, very successful for UW, which was wonderful for Dr. Belzer and Jim Southard. And for me, it was quite nice as well, because I had a successful PhD. Back from the US after my clinical fellowship in medicine, I was able to get a post at the University Medical Center in Groningen as head of abdominal and transplant surgery. And together with colleagues, we set up a preservation and perfusion laboratory. Of course, in those days, the complexity of donors had increased, and also their age. Much older donors were now used because basically the gap between the number of donors available and the organs needed was too big. The question, however, is, were we actually doing the preservation, the best preservation that was possible or not? So we just wondered whether cold storage preservation with those more complex donor organs was sufficient, and thought, well, maybe we have to revisit machine preservation. So when the LifePort came to the market by Organ Recovery Systems in the US, we thought, why not get the device to Europe and do a European machine preservation trial in kidney transplantation in close collaboration with Eurotransplant, with two very keen and brilliant young medical students who were doing their PhDs, Cyril Moers and Hugo Maathuis. We actually did 3 separate randomized controlled trials, an overall study, and then one focusing on DCD, donation and transplantation, and the other one on extended criteria donation. And what we saw is, in the end, is that as an independent variable, machine preservation was significantly better than cold storage as far as delayed graft function was concerned, in a multivariate regression model, together, of course, with a number of other already known factors which were better. At the same time, which we had not anticipated, we saw a significant better one-year graft survival, both in DBD and DCD donor organs. You see here, if there's no delayed graft function, machine preservation and cold storage are quite equal as far as survival is concerned. However, as soon as there's delayed graft function, and we all know that we can't predict which kidney is going to have a DGF, there was a significant improved preservation, improved survival after machine preservation. A follow-up study was done to detect the DCD success. We looked at renal resistance. We looked also in extended criteria donor, and again, machine preservation had better results as far as DGF and survival. We also looked at cost effectiveness, because health authorities, of course, also want to know whether a new strategy provides better health care. And indeed, machine preservation, despite the fact that you have a device which is depreciated and consumables, because of the reduction in DGF and the survival, was very much cost effective. This is a slide which we recently submitted to the New England, and it's now in press, where we did actually a 10-year follow-up on this first European multi-center trial in kidney transplantation, demonstrating that still, if there is no DGF at the very beginning, survival is very much the same, even after 10 years. However, if there was a delayed graft function. Machine preservation, also long-term, is much better than cold storage, actually a bit more widening at the very end. When I was back in Groningen, there was also a unique opportunity because there was a very close collaboration of our lab and the Department of Transplant Surgery with biomedical engineering. So we thought, well, the current MP systems the devices are quite old-fashioned, roller pumps. We need something new. So together with a colleague from bioengineering and my comrade in arms in the lab, Henry Leuvenig, we submitted a grant to the Dutch NIH, which is NWO, and got a 5-year grant to improve the device. We got 2 very keen students who were able to help us and work on their PhD, and we thought, why not kick off first in liver transplantation and look after how can we maybe look at the Achilles heel in liver transplantation and reduce biliary complications. And maybe machine preservation could be a help. So we said, well, what about technology? What kind of temperature? Do we need oxygen? We want to have a portable machine, but it should also be possible to do hypothermia and normothermia. And we would like to cover both liver and kidney transplantation. Here, the 2 PhD students, we started in 2000 and delivered the first prototype in 2007. So don't forget, this kind of developments take time. You see here a nice photo of their thesis. Arjan van der Plaats, who was the engineer, and Niels Hart, who was the medical colleague. In the very beginning, we said, well, if we want to do, for example, perfusion, we have to know about flow and pressure. So let's look it up. Well, the literature didn't reveal anything about that. So we had to go back to the bench and say, well, what kind of pressure and flow do we need? And what is tolerated under cold conditions? So we first started, for example, with 50 millimeters of mercury, looking at acridin orange, which shows in the liver a beautiful perfusion, as you can see, bright and green. However, when we did an exclusion dye at the same time using propidium iodide, which demonstrates dead cells, we saw that at that pressure, about 60% of all endothelial cells were dying, which obviously is something you don't want to happen. So then we had to reduce further down and ended up as 25 millimeters of mercury, having still complete perfusion. However, as you can see, no dead cells and an intact endothelium. We also looked, for example, what about liver oxygenation in the cold? Is it dangerous or is it helpful? And there was no increase in precursors of oxygen for radical injury at 21% or even 95% oxygen. And what we also saw is that in ATP regeneration, the oxygenation was very helpful in the liver, and as we saw later, also in the kidney. Now, when we had developed this Groningen prototype of the hypothermic machine preservation system, we had to test it in pig liver transplant. So we went to Barcelona because our colleagues Valdecasas and Fondevilla in Barcelona had a wonderful liver transplant model in the pig, which is quite sensitive and quite difficult. So not many centers in those days had this wonderful model, and we could test the machine preservation there for the first time. We also looked, for example, at the oxygenation of kidney perfusion in a DCD model, 60 minutes of warm ischemia, and then kidney transplantation to pig. Again, also confirming that in kidney transplantation, oxygenation during machine preservation is helpful. Here you see, for example, the first prototype for the portable kidney. Of course, surgeons like to stick with tradition, so we kept the box intact. We added the ice, and in the ice you see the prototype of the technique with the cassette, the oxygenator, and then on the side the oxygen supply and the engine of the pump. Normothermic machine preservation obviously is part of the Holy Grail, and it recreates a physiological environment. So we also wanted to see whether with the same device, but some alterations, we could add normothermic preservation as well. Because here you deliver oxygen, you have a physiological temperature, and you provide nutrients, which hopefully kind of initiates repair of donor organs. when we had the first trials of this, and you see here a photo of the first NMP device, we went back with this prototype again to our colleagues in Barcelona to do pig liver transplantations. And as you may be aware, in Spain, there's a long tradition of uncontrolled DCD type donors, which are basically individuals who suffer a cardiac arrest outside the hospital, are then resuscitated, unfortunately unsuccessful, but then it is still possible to obtain the organs, which is a, let's say, high warm ischemia type model. We went to Barcelona and tested this again in the liver transplant model, a DCD model with cardiac arrest for 90 minutes, typical Spanish. We tried to kind of convince them, let's carefully start with 45 minutes or maybe 60. No way. They said, listen, we have to be very practical. We go to 90 minutes warm ischemia time, which we did. And you see here that if there was just cold storage, after 90 minutes of warm ischemia and a bit of cold storage hours, like 4 hours, no animal survived. In those days, you could still do survival experiments, which obviously now is not allowed anymore. Then we added 1 hour of warm perfusion using this device, and then still added 4 hours of cold storage. And suddenly we saw an increase to 53% survival. And if we actually continued the warm perfusion for 1 hour plus 4 hours ex situ, we get 85%, demonstrating that it is possible with normothermic machine preservation to have a high graft survival rate with this prototype. Following all these wonderful and exciting experiments, we formed a spin-out company, and a family of devices was developed, which was named OrganAssist and later acquired by XVIVO, and is now a part of the XVIVO perfusion family with new prototypes with miniaturized technology, pulsatile, adjustable flow, and oxygenation for both hypothermic and normothermic use. In 2011, I got this wonderful invitation to cross the channel and swim to England, a new post and an exciting next step in my career with 50% clinical and 50% research, which later on became more research and less clinical as maturity goes on at the Nuffield Department of Surgical Sciences. Situation in the UK, the same as on mainland Europe, a lot of uncertainty at time of offering. Is this a good organ for my recipient? So many organs are declined and not retrieved or transplanted. What do we know actually of the quality of donor organs? Well, we have a lot of clinical parameters, but unfortunately, many of those clinical parameters are just not sensitive enough. So we recognize in a way a footprint, but what we need is a fingerprint. Reason for us to start in the UK as a consortium of academic centers together with our sponsor, NHS Blood and Transplant, the so-called Quality in Organ Donation Biobank, which is a national biobank and scientific platform with integrated clinical data in organ donation and transplantation. Here you see some of the members of the core team, and of course many more members outside in the hubs in the country, trying to collect, process, and then store those organs for further research. With this biobank, which has been now in existence for more than 10 years and collected more than 8,000 donors and 150,000 samples, we have received better insight in mechanisms and biomarkers using highly sensitive and novel techniques to stratify risk, see how we could correlate with good outcomes or bad outcomes using, for example, transcriptome, proteome, or metabolome, or just very plain histology. When I came to England, I also started to collaborate with my partner, Peter Friend, who actually had set up already in Cambridge with his colleague Constantine Cusius, who is an engineer, a new device for normothermic liver preservation. And when in Oxford this came to a conclusion and a wonderful new exciting machine was developed with a spin-out company called Organox, the Mitra, and the first clinical trial was done with King's College London and the QE in Birmingham to demonstrate that prolonged long-term normothermic machine preservation of the liver was very feasible. So Peter and I then thought, well, with all these difficult types of donors like ECD, DCD, type 2, type 3. What shall we do? Shall we just improve preservation solutions? Should we go for cold? Should we go for cold with oxygen or warm? Why not see whether we can do a strategy? So we went and built a consortium for organ preservation in Europe, including here, as you see on the slide, a number of partner centers, and with those partner centers, a lot of other colleague transplant centers in all those countries involved. We went to the European 7th framework, nowadays Horizon network, and applied and received 7 million euros to do clinical trials. So this is really a wonderful collaboration where we actually did 3 clinical trials, a liver trial on normothermic machine preservation versus static cold storage, and 2 kidney trials where we looked at static cold storage with machine pumping at the very end or continuous pumping at the beginning versus with oxygen versus pumping without oxygen. Now, as far as the clinical liver trial, you see here that which was headed by Peter Friend, and the PhD student was Dave Nasrallah. We saw that there was a very successful long preservation with better function and high utilization. The conclusion was that prolonged NMP for the liver is safe. It allows time to assess and recondition, especially in high-risk livers. As far as the clinical work in kidney transplantation, we first looked at the pump trial, which is now an extended criteria donor, simple cold storage with cold storage plus machine preservation in the cold with oxygen at the end, like it is done in many centers now in the UK, but also in the US. No significant difference in functional survival, basically telling us that after cold storage, a brief period of only, let's say, 4 to 5 hours of machine preservation is just not sufficient. Our primary trial, the Eurotransplant trial, which I told you about, had about 10 to 11 hours machine preservation. It was very successful. We then continued with another trial where we now compared machine preservation in DCD, also a higher risk group, continues from the donor hospitals to the recipient implantation with oxygen or without oxygen, thus a much more prolonged preservation period, a paired trial demonstrating again better function, 50% lower rejection, and a higher survival when oxygen was used versus standard. So what about normothermic preservation then of the kidney? Well, wonderful work has been done by Mike Nicholson and Sarah Husgood in Cambridge, first in Leicester and then finally in Cambridge, who, after a lot of experimental work, then went to the first clinical study demonstrating that NMP for kidney transplantation was feasible and safe. They then also did their first clinical trial of cold storage, followed by 1 hour of NMP and then transplantation with a primary endpoint of delayed graft function, which was feasible and safe, however, did not show a difference. I think we have now all reached the moment that we see that we need to prolong NMP of donor kidneys, for example, to a few hours, 6 hours, or even longer, to test. And in this kind of scheme of, we had a new device developed by Organox in Oxford, where Richard Dumble, our PhD student, following the work by Annemarie Weissenbacher, said, well, let's try to kind of prolong normothermic preservation. And what Richard did is he did a phase 1 clinical trial to assess the safety and feasibility. 36 DBD and DCD kidneys were included. Preservation up to 24 hours, including end of NMP, with a minimum of 2 hours and a maximum of 23 hours. All organs were transplanted and functioned, no primary non-function. DGF in both arms was the same, but obviously this was a safety trial and not a comparison. So I think it's fair to say that prolonged NMP after cold storage is feasible and safe. In this context, James Hunter, who's leading on our perfusion research in the lab, said, well, it's not just about resuscitation, but we need targeted intervention. So we set up a number of models, either using organs from the abattoir, from the animal house, or declined human organs, to then basically look at basic science aspects or translational research, and then finally end up in clinical trials with a lot of scientists involved as well as PhD students focusing on certain either assessment or topics of target intervention. One of the target intervention, for example, is done by John Fallon, who's one of our research fellows looking at complement inhibition and thrombolysis doing NMP. We know that complement can be very devastating to organs, especially to the kidney, but also other organs. And why not target during machine preservation and find that this complement aggregation is just not happening and immunogenicity of the organ is reduced? Or, for example, another study by Carlos Herreza and Hussein Abbas, supervised by Peter Friend, where now liver preservation is done normothermically in very fat livers, as you can see, with defatting agents, then demonstrating that after transplantation, these organs are successfully working without complications or primary non-function. The pancreas has been a bit the orphan in preservation research, so it was very good to see that James Hunter and Mo Al-Zawari are now focusing on a study where pancreas will be oxygenated and machine perfused in the cold and then transplanted in Oxford to see whether this is safe and feasible with less edema and hopefully a reduction in pancreatitis. At the same time, then finally, we also looked at the mechanism of tissue injury and repair. What is the effect of machine perfusion, for example, on the tissue proteome? Letitia Lofaro is one of our senior scientists in the lab, looks, for example, at this translational work. She has done in all the livers of those clinical trial proteomics, then trying to find out how much is protein translation, what is the endosomal pathways, and what's the role of mitochondria. While, for example, Maria Kaiser, who's one of our other senior scientists in the lab, looks at identification of biomarkers of donor organ quality, and can we maybe predict which outcome of the kidney transplantation will happen by already assessing either factors in the donor or during perfusion. What is the immediate future then? Well, I think in the UK it is very much establishment of national units, which we call ARCs, or assessment and recovery centers, where we can assess and resuscitate organs. Are they viable or not? Can we condition and repair, either using pharmacological agents or cell therapy, and initiate maybe regeneration, which of course would be wonderful if we can do this by either genetic or immunomodulation. In fact, making untransplantable organs transplantable. Now, this is a beautiful photo of the Groningen ARC, which is in the Netherlands and has been functioning for years now. In Oxford, we're slightly more modest. NHS has, of course, less finances, but we're starting nevertheless. You see here two of our fellows, Fungai Dengo and Richard Dumbill, in our perfusion lab with two human organs, which are declined organs, liver and kidney, on devices. Finally, then, after all this curiosity, commitment, and collaboration, this is only possible, this work, by all people involved, and I really would like to thank them for their support they've given me over the last years. And what have I learned from my academic journey as a final, let's say, grey-haired individual, which I'm able to say? Well, to remain curious and to be creative is very important, because it will result in innovation and relevant work benefiting patients. I think it's also important for all our young and brilliant academic surgeons to commit yourself and lead by example. You will gain trust from colleagues and patients, and I think this will allow you to really deliver. And very important, and I can't underpin this too much less, is collaborate. Standalone unicorn research often fails. Sharing ideas and responsibilities leads to high quality and wonderful studies. And last but not least, don't forget to have fun, and I wish you all a lot of success. Thank you very much.